Good afternoon. Today I will be presenting part of my master thesis, which focused on speciation in mound-building woodlands. Speciation is the process by which emergent taxa accumulate differences over time until reproductive isolation is achieved and new species are formed, represented here by species A and B. Reconstructing divergence histories using genetic data has become the gold standard in speciation genomics, and this is usually done by sampling a single population of each of the species under study. But as species are distributed over physical space, heterospecific populations may relate to each other differently in the spatial context, which can impact the inference of divergence histories. Let's assume, for example, that species A and B diverge without gene flow, and that presently the populations A2 and B3 exist in a lopetry, and that the populations A3 and B1 are currently exchanging migrants. If we were to reconstruct the speciation history between species A and B with populations A2 and B3, we would obtain a population history that matches the history of the species. But if we were to use populations A3 and B1, the history between these populations would not match the overall species history. And so we performed multiple population comparisons to understand the effect of the spatial context of populations on their inferred divergence histories. We used mound-building redwoodlands to do this, particularly the species Formica polyctena and Formica aquilonia. They have contrasting distributions in Europe, overlapping in limited areas of the range, and present-day gene flow is spatially heterogeneous, as the species are known to hybridize in Finland, but hybrids have not been recorded elsewhere. Using these ends as a model allowed us to not only test how the inferred divergence histories may vary depending on the geographic locations of the populations, but also to elucidate the history between these hybridizing species. We used populations sampled at different points of the European ranges of the species and paired them together in 4 comparisons. And then we reconstructed the divergence histories between these pairs of populations. The first 2 pairs compared the F. polyctena population sampled in Switzerland to the F. aquilonia population sampled in Scotland, and these pairings are presently allopatric. We then have a comparison between Swiss populations, which may be presently parapatric. Our last pair compares the Finnish populations of these species, which are known to be sympatric. 3 females were sampled from each location outside of Finland, and 4 females were collected from the Finnish populations, to a total of 20 individuals. We obtained high-quality genomic data from these samples via paired whole-genome sequencing, and after SNP calling and filtering, we had over 2 million sites, from which we built site frequency spectra for each pair of populations. And this is an effective way of summarizing the distribution of allelic frequencies in the populations. All of these data sets add over 500,000 SNPs. We reconstructed the demographic history of the 4 population pairs with FASTMCoL2, which is a coalescent SFS-based method, meaning that it tracks the ancestry of lineages backwards in time until the most recent common ancestor is found. We tested 8 models that considered all plausible scenarios of divergence between these species, including the possibility of introgression from an unsampled species. After testing all models with all population comparisons, we found that the overall speciation history is consistent across all pairs. And this history contemplates a scenario of divergence with continuous asymmetric gene flow. After divergence, migrants flow only from the F. aquilonia population into the F. polyctena population. And that then at the time of the size change, when the populations are allowed to either expand or contract, migration becomes bidirectional. as both populations are allowed to send migrants into the other. Looking at delta likelihoods of the 4 best models, we see that this model, highlighted in green, is clearly the best across comparisons, as it has the lowest delta likelihood. The delta likelihood is the difference between the expected likelihood of a model and the highest possible likelihood we could achieve. So the lower the number, the better the model is. And while the overall divergence scenario is common to all comparisons, we still found variation in parameter estimates between comparisons. With the best histories that we inferred for each of the population comparisons, we can pick out the similarities and the differences in parameter estimates. We're going to take a look at figures like this now, where the populations are represented by boxes and the times are represented in number of generations. The effective sizes of the populations are given in number of haploids, and the migration rates are given in number of migrants per generation. Looking at the best histories, we can see that the times of divergence are quite consistent across all comparisons. We consider the generation time to be 2.5 years, which puts the divergence time in the Pleistocene between 528 and 561,000 years ago. We can also see that the estimates of the ancestral population effective sizes are very similar, both for the ancestral populations of the species and for the ancestral populations of each of the sampled populations. And finally, the migration rates between ancestral populations are also very similar across comparisons. And looking at these parameters that are similar, we can see that they pertain to older events. So we suggest that they are similar across comparisons due to shared ancestry, or in other words, because the sample populations of each species share the same ancestral population. Okay, now we can identify the differences between comparisons. Both the estimated times of the size change and the recent population effective sizes are vary greatly across the comparisons, which may be derived from the independent evolution of the sample populations in recent times after splitting from their ancestral populations. The most notable difference we found across comparisons is in the estimated rates of contemporary migration. These differences match the present-day context of the populations we paired together, as we can see that for the first 2 comparisons, where the populations are presently allopatric, we inferred no recent gene flow between the populations. Then, for the 3rd comparison, as seen here, where the populations may be parapatric, we inferred recent gene flow to be reduced, and but still happen unidirectionally from the F. aquilani population into the F. pollicrana population. And then lastly, for the Finnish comparison, where these populations are known to hybridize, so they are sympatric, migration not only increased, but it became bidirectional, as we now see migrants moving from the Finnish F. pollicrana population into the Finnish F. aquilani population. And so we can see here that if we had used only the Finnish populations, which are of greater interest to us, we would have obtained and accepted a divergence scenario that is not representative of the species history. In this study, the differences in the geographic context of the populations caused variation in the estimates of recent demographic parameters. But there are other plausible scenarios that could create variation in parameter estimates, such as utilization of different refugia during glacial periods, secondary contact with a 3rd species in a limited region of the range, or low effective sizes. We suggest that when inferring divergence histories, it may be beneficial to sample more locations while sampling less individuals per location in order to obtain an accurate picture of past and recent events. The work I just presented is part of our paper, which is now available in bioRxiv if you would like to take a look. I would like to thank Vítor, Sousa, Jonna, Kulmuni, and Pierre Nouhaud, who supervised my work and are senior authors on the paper. and also the co-authors that did the sampling and the morphological identification of the samples. Thank you very much for your attention.